Hey team, so we're going to take a look at an NHT biology question today. It's not the expected format that we always see on VCAR exams. However, I'll give you a few tips here to work through it so that we can tackle what is maybe an otherwise confronting question. All right, guys, so here it is. This is question 6 from the 2023 NHT biology exam. And it's a standalone question here, so it's just this one 4-mark question. You can see there's a big diagram here for us to have a look at. It's probably actually a microscope image. So the human immune system has many different types of cells. As we're probably well aware, we need to know all of these cells and be able to make comparisons between them. The diagram below illustrates 2 of these cell types and their interaction with each other. So we've got here that they're interacting with each other, which will maybe be a big giveaway for us here. Cell A, right, so we've got cell A here is adaptive. So we're thinking cell mediated or we're thinking humoral, one of the two. Cell B is innate. It also tells us here that cell B has an antigen attached to its plasma membrane, which potentially suggests that it has an MHC2 marker. There's a few pieces of information there that I think are going to help us. If we scroll down a little bit, I might just zoom this out a touch so we can see it. What we've got here is these 2 cells, right? We can see this cell B here, which we know is a part of the innate immune system, and it possibly has an MHC2 marker as it's presenting an antigen. And it looks like it's got these weird sort of little tentacles coming off it, which, if we're familiar, might look like some dendrites. Okay, so it's probably suggesting to me that this is a dendritic cell and that it's presenting something to this other cell here, cell A, which is adaptive, right? If we know sort of our steps of the cell-mediated or the humoral response, hopefully we're thinking that this might be a T helper cell, and this is probably some sort of APC. It's probably potentially a dendritic cell. Great. So what we've got is a T helper and a dendritic cell. Hopefully we potentially don't even need to say this is a dendritic cell. I can't remember off the top of my head if it was accepted as just an antigen-presenting cell or not, or just a macrophage or a phagocyte. Not sure what they actually accepted in 2023 off the top of my head. But from looking at it, it has these dendrites. We might be able to tell it's a dendritic cell. If we know the specific cell type, it's probably better to say what it is specifically. This is just a 4-mark question that tells us at the top. There's no mark allocation here, right? So don't get thrown by that. If you have these standalone questions in VCAR exams, you're only going to know the mark from what's up here. So it's a 4-mark question asking us to outline the events in an immune response, which will occur after the interaction between these 2 cells. And again, the 2 cells, we've got a cell A and B, and it's saying what will occur after the interaction. So after the interaction, we're saying that the APC is presented to the T helper cell, to dendritic cell. Okay, so we might be saying what happens after. We can't be talking about the presentation there to the T helper. We might be saying that the T helper releases cytokines to a selected B cell. Okay, we might start talking about the humoral. We don't necessarily have to talk about humoral here. But we could selected B cell. Cool. That's one possibility. We could also be stating here that the B cell undergoes clonal expansion and differentiation. It's always sort of nice to add more information if we can. So we're going to say what they differentiate into. which are going to be our plasma cells, and our memory cells. Great. We can just keep going down the line here. Again, it's 4 marks. We've got 2 marks here. We're going to be saying next, what happens next is what occurs after the interaction, remembering. So it's plasma cells produce antibodies. And whenever we're talking about antibodies, it's usually nice here to mention that they're going to be specific to the antigen presented on the APC, the dendritic cell in this case. Great, so we've got 3 marks here. Again, doesn't tell us it's 4 marks. We're going to have to look at the top there and say that it's 4 marks. We could then potentially go on here to say the function of the antibody. So we might be saying here that the antibodies, the immunoglobulins, agglutinate the pathogen, the specific pathogen. Right, we might also be going on here potentially to saying that the memory cells provide immunological memory. Right, all of these things are happening after the interaction. Right, we've gone down the road of doing the humoral response here. There's no reason necessarily why you can't go down and do the cell-mediated, right, and say the T helper cell activates cytotoxic T cells, then describe that a cytotoxic T cell will go and survey infected cells that are presenting that specific antigen on their MHC1 marker. Right, we've just gone this way. You don't necessarily have to do that. If you've attempted this question and you've done cell-mediated and you've got 4 distinct points for your 4 marks, then that's great as well. Hope it's been of some help to you. When we get these questions, right, trying to be overwhelmed by these electron microscope images here, right, read through the information at the top, even if it is just a few sentences, because there's some really key information in there that is going to point you in the right direction of what these cells are. Even if we don't recognize these cells straight off the top of our heads, the information at the top there provides a little bit of insight that's going to aid you in responding to the question below. Cheers.